Welcome to the Spring 2021 Core Facility Seminar Series. Today, we will be featuring the flow cytometry facility. For those of you who do not know me, I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. In today's seminar, you will hear from the director, Amy Burnside, who offers expertise in flow cytometry analysis and cell sorting, as well as high throughput imaging. We have two guest speakers, Brian Rosario and Gary Cuneo, both from Luminex. We hope that with these bi-weekly seminars, you will discover what great resources that the centralized UMass Amherst core facilities offer to our campus community, the New England region, and beyond. Next, just a few housekeeping items. This seminar is being recorded. If you miss any part of this seminar or would like to forward it to someone who could not attend, there will be a replay of this and all our previous seminars on our website. I'll put the link in the chat. I recommend you set your view mode to speaker. Please stay muted during the talks. We will save the Q&A until the end of the presentation. During that time, I welcome you to unmute yourself and ask your question. So now I'd like to introduce you to Andrew Bernard, the Director of our UMass Amherst core facilities. Andrew?
Thank you, Lisa. Good afternoon, everyone, and thank you for joining us today for the third core seminar of the spring semester. As Lisa mentioned, my name is Andrew Bernard, and I'm the Director of the Centralized Core Facilities here at UMass Amherst. Our state-of-the-art labs, instrumentation, equipment, and world-class experts are helping researchers drive discovery and pursue scientific endeavors across all disciplines. The cores are open to anyone from students through senior scientists, regardless of their affiliation, including researchers from other academic institutions and commercial partners. To help access our cores, there are several funding opportunities. For IELTS investigators, core credits and center seed funding is available. For our external users, the Mass Innovation Voucher Program subsidizes usage for small companies based in Massachusetts at up to 75%. This program has awarded more than 500 vouchers, representing more than $5 million in total project costs across the 5 UMass campuses. We aim to be your partner by expanding your research capabilities and productivity. If you're a UMass undergrad or work with UMass undergrads, we are again hosting the Core Summer Internship program this coming summer. The CSI program aims to provide STEM students hands-on experiential research and technical training in the core facilities. You can learn more about the CSI program at our upcoming overview and Q&A session next Thursday, March 4th at 3 p.m. Registration and additional details are available on our website, which we will put in the chat. Please feel free to reach out to me or any of the core facility directors if you have any questions or would like more information, whether it be about what services we have to offer or getting access to the facilities or training. It's now my pleasure to introduce Dr. Amy Burnside, Director of both flow cytometry and animal imaging cores. Dr. Burnside joined UMass in 2008, taking over management of the flow cytometry core in 2011, 5 years before IELTS or centralized cores were a thing here at UMass Amherst. Her experience as an educator and her in-depth knowledge across many disciplines make her an indispensable component of the research enterprise. We are incredibly grateful for Amy's contributions to the core facility and research communities here at UMass. With that, I'll pass it to Amy and I'll make myself available during the Q&A. Thank you.
Thank you, Andrew. Thank you, everyone, for taking the time out of your day to join us. The flow cytometry facility has 4 pieces of instrumentation. We have a Fortessa, dual Fortessa that is available for flow cytometry acquisition analysis. We also have a ARIA 2 that is being replaced actually this year. That's capable of fluorescence-assisted cell sorting. And what we'll be talking about today is our imaging flow cytometer, which is an Amnis ImageStream. And we also help with flow cytometry education, and I can help with experiment and research design consultations, as well as help you with your grants and give preliminary data or letters of support. Again, I've mentioned our tools, but we have 2 dual Fortessa. Affects ARIA cell sorter, and here are some pictures from our Amnis ImageStream showing nuclear versus cytoplasmic expression, and here we have colocalization. If you're working with primary cell lines and would like to monitor growth, death, or cell cycle, we can help you with that. If you would like to characterize your immune response or tumor cells, one of the biggest projects now are people using CRISPR-Cas9s or other forms of transgenic cell lines, and we can help you sort and create your transgenic cell lines. So reach out to me if you have any questions about what we offer. But today we'll be talking about our Amnis. There's my background, our bio slide. And then I'd like to introduce Brian Rosario, who's our Amnis specialist from Luminex. Brian's from Brian is the Amnis sales specialist, has been with Amnis and Luminex since December last year. He holds a Bachelor of Science from the Bryant University and is a major in marketing and minor in communications. And he was an advanced surgical sales specialist with ConMed Corporation. After Brian, Gary will be joining us, and he's our Northeast Field Application Scientist and has been with Luminex since October 2018. He holds a Bachelor of Biology from Merrimack and a postgraduate at Tufts University in Cell and Molecular Biology and Harvard University in Engineering Sciences. In his previous role, he was a flow cytometry and imaging core manager at the Weiss Institute for Biologically Inspired Engineering at Harvard University. Thank you, Brian and Gary, for joining us today. Brian, if you'd like to take over screen sharing, I'll hand it over to you.
Great, thank you. Okay, is that working? Everyone can see my screen?
That looks great, Brian. Thank
Great, thank you. Well, thank you very much, everyone, for spending some time this afternoon with both Gary and I. We're just going to do a high-level overview of the Amnis system and the software. Yeah, questions, as Amy had mentioned, can be saved until later, unless you feel that you know, something needs to be answered right away. Feel free to throw it in the chat, and you know, we'll try to get those answered as quickly as possible. Some of what I'm going to be going over is going to sound, for lack of a better term, canned. The reason being is that there's some legal things that I have to literally read. So if it sounds like I am reading, it's in fact because I am reading. But it's going to be pretty casual. So we're just going to jump right into it here. Just an agenda, we're going to go over the Amnis system, how it works, how to get the most of your current imaging flow cytometer, how the software simplifies the analysis, and if we have time, any available options and upgrades. But you guys have a pretty thorough system over there so far. So Amnis pioneered imaging flow cytometry. Imaging and flow combines the techniques of imaging with those of flow cytometry. So throughout this presentation, I'll aim to demonstrate how this unique combination can help you design an informative experiment and with greater confidence in the results. So as a cell biologist, you're interested in understanding the process going on within the cells under normal conditions and disease. So to study your cells of interest, you might ask questions about the number or percentage of those cells in a mixed population. You might ask questions about the organelles, the nucleic acids, or about different proteins and molecules at the cell surface or inside the cell, and the way those might be different under different biological conditions. Today, when addressing some of those questions, researchers typically turn to flow cytometry or microscopy. Flow cytometry provides the information about molecule expression and population statistics and sometimes activation status. Because of the high per cell throughput, this provides objective data with strong statistical power, even for rare populations. So all this information provided through this method, though, is simply a measurement of fluorescence and changes in that fluorescence. That is the foundation of all the information provided. When asking questions about the size or the shape of the cells, where in different cell, where in the cell different molecules are located, or how cells might be interacting with one another, as well as the appearance of the cells, one turns to fluorescence microscopy. Where in a cell a signal is collected allows for the gathering of deeper information, but sample size is often limited when utilizing microscopy. As separate technologies, both are powerful and definitely have their place, there's no question about that, but with Amnis, these tools are combined into a single tool that takes advantage of those strengths and extends them while allowing those measurements to be derived from the same cell, which is unique to this platform. The results are greater than the two individual parts. This allows for deeper biological insights, for a higher quality, statistically relevant data in less time. It allows you to make novel discoveries, learn mechanisms of action, and you're confident that the science is done properly and that it's verifiable. What we invite you to do is allow your science to be about that new insight and technique you can contribute to your field instead of just repeating the same old experiments with the slightest of variation. So the next few slides will break down these points a bit, starting with the power of contextual verifiable results and how this leads to getting more from every experiment. So what information does imaging cytometry provide that flow cytometry and microscopy can't provide on their own? Well, first, imaging flow cytometry uniquely allows for simultaneous data collection. Because of the multispectral imaging capabilities, you collect expression information, such as cell surface biomarker expression that you get from traditional flow cytometry, but within those very same cells, you can add contextual information. Ask your questions like, are these two molecules co-localizing in one population but not the other? This is the only way directly correlative data can be obtained. It is like a virtual sort. If you need information about both, why not do it at the same time from the same experiment from the same cells? Secondly, those results are immediately verifiable. It can be validated through image analysis. If you've ever struggled with image data or how to relate the images taken on a scope to the greater population, this platform provides that solution. You can demonstrate the significance of any image to the population directly. In the second panel, you see that the cells have internalized one particle represent 50% of the overall population. Visualization at the population level will confirm and demonstrate statistical significance and relevance of your conclusions. Conversely, you can expect any data point in a dot plot and determine whether it's important or not. You can verify if it's a fact of bona fide double positive event or doublets, or including for rare artifacts. The third, the combination of these technologies allows you to collect highly feature-rich data that can be used in analysis. By its very design, the Amnis system is in a unique position within the context of other individual platforms. Microscopy is often limited in speed and the number of fluorochromes at a time. Flow cytometry has the throughput, but it's very feature thin. With Amnis, we're able to capture both those features there. So what we're asking is, don't limit yourself in your experiments and don't data dump collectible and valuable information down the drain. Now I'd like to talk about the combination of these two technologies also represents a significant savings of time. Right? First, as with traditional flow cytometry, cells move through a flow in an Amnis system are directed toward interrogation point. Interrogation is rapid and automated using familiar tools such as user-defined gates for collection and counting of specific populations. By automating the imaging process using a fluidic architecture and traditional flow-based gating strategies, you're able to confidently collect as much as 1 million images per minute with unbiased sampling and giving strong statistics and consistency across multiple experiments, even for rare events. You're shown up here. And am I frozen? I think my computer just froze. See if I'm able to go back. There we go. Just a moment. Let me see if this unfreezes here.
Yeah, we can see all your notes right now.
Let me technical difficulty. Hold on one moment.
I have the presentation as well if you need me to pull it up.
Yeah, let me just see if I can bring it up here. My apologies here. Let me see. Here we go. How's that, Gary?
We can see it.
Can you see my notes?
No.
Okay. All right, let me get back to where we were here. All right. So thank you for that. My apologies. So where was I? So secondly, so with the imaging and flow, it allows for collection of data for non-inherent cells in objects such as immune cells and marine organisms, bacteria, virus EVs, other particles without the need for spinning onto slides or fixing to a matrix, which may lead to artificial morphologies. See, there we go. Beautiful. Third, the imaging and flow produces data that is in a perfect format to benefit from classic and cutting edge analysis tools. Our Ideas image analysis software provides these classical tools and now also offers optimized machine learning and artificial intelligence data analysis, something that Gary can go over a little bit later. So the nature of image collection and flow creates a consistent background leading to easy image segmentation. This, coupled with the nascent machine learning and AI tools, greatly accelerates and improves data analysis, especially for things like difficult morphometric analysis as shown here. So in summary, the combination of flow cytometry and imaging within one platform creates a new path to novel discovery, and it enables you to design experiments that efficiently and objectively consider the mechanisms of action and processes in cell biology relevant to your research. The statistically strong and visual verifiable data reaches a high standard for scientific rigor and excellence. There are unlimited ways that this can be applied. And I imagine that being able to quantify things like receptor internalization and trafficking and cell-to-cell interactions, and the activation of signaling cascades, and then connect them with how they may ultimately lead to something like nuclear translocation of transcription factors in gene activation would be of high value. Now imagine this could all be done under one platform and many within the same experiment. So that's exactly what Amnis offers. By being able to look at many of these aspects and applications together, confidence in your results are strengthened and deepened understanding is achieved, all with higher likelihood of getting the science right the first time and in less time. And it does set a new standard for scientific discovery. So what does the new standard mean? It differentiates and elevates the impact of your research. Here are just some of the high impact factor publications that have showcased the capabilities of the Amnis platforms. Many researchers have shared with both Gary and I that their Amnis data was important in part of getting their paper accepted, especially to a higher tier journal or getting a grant funded. We encourage you not to miss out and discover the full advantages of that come with the Amnis imaging platform that's available at UMass.
So I think we can open it up for questions at this point.
I know we have one advanced question. Gary, did you see the email? I forwarded it to you. It was about asymmetrical division.
Yeah, I had responded, I believe, to you just
Okay.
based upon publication. So Andy Philby in the UK, he's also one of the ISAC's emerging leaders. He previously was. He has done some research in that area. What we've found is if you don't have a good probe for measuring that, it can really affect your experiments. I did a quick search and a lot of them are having trouble just finding good fluorescent ways of probing those to get
Mhm.
results. It is possible, but it does require a little bit of work on the experimental design for things. It's not, but basically once you get it working, it's easy for us to quantify it and measure it.
Right, okay. If there's any other questions, please
Yeah, I
feel free
see.
to.
I see one other question, and the question
Okay.
was, are the images flattened, or are we looking through one focal plane? So the images are not flattened. The flow cell itself has a 250 micron diameter. These cells are in suspension, and the polystyrene beads are there to focus. There's a lot of the images I showed, they were at 60x, which means we have a 2.5 micron focal depth. So everything within there is in focus. Essentially, it's taking a small slice of the middle of the cell is what it's doing. It's not confocal by any means. It's not optical sectioning, but because our focal plane is so clean, you can see directly into the middle of the cell. Whereas with a lot of those, if you had taken that image at say 20x, where your focal depth is now 10 micron, you may or may not be able to see the nucleus that is within the membrane stain itself. And so, I mean, dealing with that, it's not, yeah, feel free. Michelle's asking questions.
Sorry, Amy. Sorry, Gary. So,
No
No
worries.
problem. Thank you.
so I have a question about this. Amy, as you know, our cells are a bit smaller than standard, mammalian cells.
Mhm.
And so I'm asking that particular question because depending on how our cells would be in solution when it's in front of the camera, if that slice is so thin, we could be looking at just the outer edge of, let's say, the cell membrane or something like that. Or so how would it, the question is, how does the Amnis handle that situation?
So typically so the polystyrene, so I guess we should start off with how the instrument works. So you have the polystyrene beads, and they're being focused by hydrodynamic focusing. So just like a traditional cytometer, we're still focusing those into a core stream in a single cell state. Those polystyrene beads focus on the middle of the flow core, and that's where our objective is focused. So
Okay.
typically you won't just see the outside of the cell, you'll get a really blurry bright field image if it's not in focus. And that's where that gradient RMS feature comes in handy. If you have a blurry image, you just eliminate those from the analysis. Now, even with RBC cells and platelets, I did show some of that data at 60x. Those are pretty big. They're 1 to 2 microns. So I'm not quite sure how much, bigger or smaller your cells are in relation to that. But we do get really good resolution even off of small cells. Now, if you're talking nanometers, yeah, you're just going to see a pixel.
I think we did run your cells, Michelle, and I think they actually looked pretty good. We could even see the micronuclei.
Is that so? Are you doing tox studies? Because micronucleus.
No. So what?
She's looking at
application.
division, I think.
Yeah, I study a parasite system, and it's the mitochondrial DNA that we were trying to track. And I do remember, Amy, that what we were seeing were just blurry cells sometimes, right? And so then we couldn't detect the mitochondrial DNA in those particular cells.
I'd have to look back at the data.
Yeah, but this increase in gain, the sensitivity, I wonder if that's going to help us out. And so could you comment on how the increase in gain might help out when I'm talking about dealing with cells on the order of about, the length of the cell is about 25 microns.
I mean, so it is like if the length is 25 microns, I don't know if you need more sensitivity. A lot of times, if you're looking for that, you're going to want a 60x objective. Like the resolution isn't going to change unless it was a brightness issue to begin with. So even with the resolution there, are you able to visually tell the phenotype, like, hey, this is a positive cell for whichever condition I'm looking for, and this one's negative? Are you able to see based upon the imagery?
As long as it's in focus, right? So there's those instances where it's not in focus because it's a long and thin cell, if that makes sense.
Yeah.
trypanosomes.
Okay, so everything would have to be in focus.
Yeah.
We typically don't analyze cells that are out of focus, but if it's something you can visually phenotype, like machine learning can give you a yes, no answer. As long as you can tell it, this is the situation I want, this is the one I don't want. The other thing is we now have an additional layer on top of that, which is AI. AI is able to figure out these more subtle morphologies. So micronucleus is somewhere where we find AI really working. It's based upon a convolutional neural network. And so it's a black box, essentially. It doesn't tell you how the analysis works, but we've found for these small morphological differences, whether they be trying to look at protein aggregates in injectables and separate that from silicone oil, or look at micronuclei when they're right next to these two big, bright, full nuclei, it's able to pull those out as long as you can tell it what it's looking for. So I think we could get you an answer. It's just getting your cells in focus.
Great. Thank you so much.
Mm-hmm.
We're at 2 past the hour, or 3 past the hour. So if people want to stay on, they're welcome to stay on.
One more question in the chat, Amy.
Okay.
Yeah, I see the question related to dendrites and axons.
Oh.
The issue with them is they like to grow out nice and long along a surface. When you put things into suspension that don't want to be in suspension, they tend to ball up. So I think you could lose some of the morphology you want to see in these cells. So for something like that, I tell you to go use a traditional microscope. I used to run an imaging core, and I would never tell anybody to take their nice, beautiful like neurons off of the plate, because you lose all that important data. So your microscope is still probably your best bet for those cells.
Well, I think that's it. Any more questions? Great. Thank you, Gary. Thank you so much.
Yeah, no problem. Thanks, everybody.
Yes, thank Gary.
thank
Thank
you
you,
all.
Brian.
And just
Thanks,
thank you all
guys.
for attending today's seminar. Throughout the series, we'll be featuring individual core facilities and their relevant technologies. Our next seminar is scheduled for Tuesday, March 9th, featuring the Genomic Resource Laboratory with Director Ravi Ranjan. So save the date and we hope to see you there. Goodbye.